The wrong head. No.
Sam doesn't know she got. Not loving. Yeah. I guess we could do that. Who would want to take? Okay.
So hi everyone, welcome to our fourth speaker session for the Intro to SLAC program, Intro to STEM program with SLAC. Today, our speaker is Ms. Ariana Peck, who is a computational biologist / biochemist who is analyzing an image of cells and molecules using cryogenic electron microscopes. So yeah, I think I hand it over to you, Ms. Peck.
Great, thanks for that introduction and for having me here today. Let me figure out how to get my screen shared. Here we go. Okay. Okay, can people see my screen? Really?
Yes.
Okay, great. Okay, so I guess, yeah, so thanks again for having me here. And I guess I wanted to start off by saying that if at any point in this talk, anyone has any questions or something's unclear, please don't hesitate to interrupt me. I'd really be happy to answer any questions about what I'm saying at any time. And so, as Neha mentioned, I'm a biochemist by training, but for the past several years, I've actually been working at the intersection between biology and physics. And specifically, what I do at SLAC is to write software for some of the biophysics experiments that we can specialize in at SLAC. And so that will be the subject of my talk today. So I guess a good place to start, I think, is to address the question of, well, what is biophysics? So biophysics is a field in which we make use of experiments that really depend heavily on kind of a detailed understanding of kind of fundamental physics and those processes, like the way that matter interacts with sources of radiation like electrons or X-rays. And we use those experiments to understand how biological molecules function. So actually one of the seminal experiments kind of early on in this field was carried out by a British chemist named Rosalind Franklin, and her portrait of her is shown in the upper left of this plot. Of this slide. And so Rosalind Franklin and her colleagues were really interested in what the structure of DNA was. This was in the 1950s, and at that time, the structure of DNA was still unknown. And so the experiment that she performed was to get a bunch of DNA molecules in parallel, as shown in the schematic down here. And then she illuminated them all with an X-ray beam. And so she knew that the atoms in the DNA molecules would scatter those X-rays. And so she placed a photographic film downstream of the scattered waves, and she was able to measure the image that's shown here. And what's shown up here in the upper left next to her portrait is actually the image that was in her lab notebook. And as you can see, there's this very distinctive pattern that she observes in the scattering experiment of the DNA with the X-rays. There's like this cross-shaped pattern that's very distinctive. And there are also these bands that are spaced kind of in particular distances from one another. And based on this pattern and other images she collected from experiments like these, she and her colleagues were able to deduce that the structure of DNA is actually a double helix, where you have these two strands of DNA that wind around each other in kind of a helical fashion. And there's a very characteristic length between kind of turns of the helix along the way. And that sort of information was really critical for us to understand how DNA is able to serve as our genetic code. And because that information was so crucial, and this experiment was all part of that, this work was awarded the Nobel Prize several years later. Unfortunately, at the time, Rosalind Franklin's contributions weren't fully recognized to this work. And so it was actually two of her male colleagues who were awarded the Nobel Prize while she was not. But in the decades that have followed, there's been increasing recognition that she played a critical role in these experiments. And so I wanted to highlight this story because I think it shows a really good representative example of kind of a very classic biophysics experiment in the field. And it also demonstrates that women have always played a critical role in advancing the field, even from kind of its early days. And so this particular experiment focused on DNA, but the majority of biophysics experiments actually focus on a different biological molecule, specifically proteins. And so the next question that's worth addressing is, why should we study proteins? What's interesting about them? And so proteins are biological molecules that are really critical to basically any cellular function that you can think of. Everything from kind of the simplest organisms like single cell bacteria or viruses, all the way to very complex organisms like us. We all depend on proteins to carry out a bunch of cellular functions and in order to survive. And so, proteins are the antibodies, for instance, that protect us from viruses like COVID. The proteins are the keratin that form our hair and our nails. They're the hemoglobin that are packed into red blood cells that transport oxygen throughout the body. And proteins are also many of the enzymes that catalyze critical chemical reactions inside cells and accelerate them so that they actually occur quickly enough to be compatible with life as we know it, with biology. Because if you just had some of these chemical reactions and the reactants just sitting in water, it would take geological times for some of these reactions to occur. So enzymes are really critical for accelerating. It's pretty shocking, but enzymes are pretty critical for accelerating these reactions to appropriate timescales. And so some of these reactions include things like photosynthesis, or a lot of metabolism requires protein enzymes. And so if you think about if you eat and you're starting to break down fats and sugars and even proteins into their constituent building blocks, all of that really depends on protein enzymes inside our bodies. And so in order to understand how these proteins are actually functioning, what we really need is a way to access their structure and also their dynamics. And so unfortunately, proteins are very small, so things like light microscopes won't be sufficient in order for us to actually kind of understand protein structure and dynamics. And so we need kind of more advanced technology to be able to image proteins to understand their structure, which means how all of the atoms are arranged relative to one another in three-dimensional space. And then also their dynamics, because as it turns out, proteins are really floppy molecules. And so some of them undergo hinge motions or kind of rotational motions. And that's really critical for some of the biology that they undergo. And so we need the technology in order to be able to see those motions and to see the structure of proteins. And so that's something that SLAC actually specializes in. And in particular, one of the methods that we're able to perform at SLAC and at many other places actually is called X-ray crystallography. And that will be the focus of my talk. And SLAC has a particularly good instrument for performing X-ray crystallography, and that's called an X-ray free electron laser. And that means that we can deliver really bright X-rays for our crystallography experiments. And so the rest of my talk will be broken down into three parts. The first part will be focused on how we deliver these ultra bright X-rays to the experiment. And then the second part will be how we deliver the protein samples to our experiment, because we need this protein sample and also the X-rays to collide. And that collision will cause all of the X-ray beam to be scattered off as waves. And we can collect that wave similar to what I mentioned with the Rosalind Franklin experiment. And we can analyze that and perform algorithms on those data in order to get back to the protein structure and also dynamics. And from there, we can actually better understand the biology of proteins. So without further ado, let's get on to discussing how X-rays are actually delivered. So what's shown in this photograph is actually an aerial view of SLAC. So SLAC is across the bay from Fremont, and what we're looking at here is kind of the Menlo Park area, if any of you are familiar with it. You can see, if for those familiar with it, this is Highway 280, which is going across right here. And then so the SLAC particle accelerator actually extends underneath Highway 280, and it extends for around 2 miles. It's a very, very long piece of equipment that goes from kind of over towards the hills, and beyond those hills you'd have the ocean, and then it extends down towards Stanford actually. And then so Stanford is off screen actually, it's kind of back behind us. And so this long particle accelerator is what's delivering our X-rays over to where the experiments actually happen. So that's the near experimental hall shown down here at the bottom right. And also the far experimental hall, which is shown at the bottom here. And so we have this particle accelerator initially, we have this particle accelerator ultimately to deliver X-rays, but initially we actually have to start with electrons in order to emit the X-rays in the first place. So step one of this entire process is to get the electrons going really crazy fast. So we want them to be going almost the speed of light by the time we're done with them. And the way that we accomplish this is by shooting them through the particle accelerator. And a good fraction of the particle accelerator actually consists of these cavities in which an electromagnetic field is applied. And so electrons are charged particles, so they'll be responsive to any sort of electromagnetic field. And that's shown schematically in this bottom right panel right here, where you can see what's shown in red is kind of an electron pulse. And then the lines in blue and orange show the electromagnetic field that's being applied inside these cavities. And so as we propel these electrons through the electromagnetic field, they'll start to speed up. And there are 2 ways that we can make them going really, really fast. The first way is that these cavities are actually constructed of an element called niobium. And niobium is critical because it's actually superconducting at really low temperatures. And so we cool these cavities down to minus 271 degrees C. So it's just 2 degrees below what's called absolute zero. It's very, very cold. And because it's superconducting, that means there's no electrical resistance. So essentially, it's almost like we're working in a frictionless environment where all of the all of this electromagnetic field will be applied to the electron pulse that's going through without any waste, without any loss of energy. And so that's the first strategy by which we get these electrons going really fast. And the second strategy is just to have all of these cavities lined up. So we have, many, many cavities all in a row, just along the kind of length of the linear accelerator. And that's really critical because, as these electrons go through cavity after cavity after cavity, they'll start going really, really fast. And so what's shown in this photo in this top panel here is actually a scientist kind of assembling one of these cavities and putting it together. And as you can see, there are many more cavities kind of down the length of the particle accelerator. So the second step, we have very, very fast electrons, but actually what we really want is very, very bright X-rays. And so the way we accomplish that kind of transformation, or at least, getting to the point of delivering X-rays, is to start to pass our electrons kind of through a different portion of the accelerator, in which there's these magnets with alternating polarity. And the effect of passing these electrons going extremely fast through all of these magnets is that they start to oscillate as they go back and forth between magnets of different polarity. Because again, remember that electrons are charged, so they'll be responsive to things like magnets. And so as you can see here, they start oscillating back and forth. It's almost like a skier who's racing through a slalom course. They're already going really, really fast, and now they have to go through this kind of slalom course back and forth. And the effect of this is to actually, as they kind of go, as they start oscillating, they'll actually start to emit X-rays. They start to emit this radiation that's called X-rays. And what's really critical is that the X-rays will actually continue going straight, because X-rays are not charged at all, so they'll be unaffected by any of the magnetic field. So the X-rays keep on going straight. The electrons have a slightly longer path because they need to oscillate back and forth. And so what happens is that the X-rays actually will start to outpace the electrons. They'll start to get ahead of them. And so they'll start to actually collide with and interact with the electrons right in front of them. And the effect of that is to actually cause more X-rays to be emitted. So what you end up getting are these very bright pulses of X-rays, kind of that are kind of going alongside these very, very fast electrons. And so at the very end of the experiment, what we really want delivered is just X-rays. We don't really care about the electrons. The electrons are just a means to an end so that we can get X-rays for our experiment. And so at the very end, there's this final magnet that actually kind of pushes the electrons away and diverts them to what's called an electron dump. And so, the electrons get like pushed away essentially to the garbage bin, and the X-rays continue moving straight forward because once again, they're uncharged, so they're unaffected by any of this magnet stuff. So they'll just continue going forward. And at this point, because of all of this bunching activity, they're really, really bright. And that's exactly what we want for our experiment. So then we can deliver all of these X-rays to our experiment, and the site where we actually perform this experiment we call a hutch. So this is the view inside one of the hutches that we actually use for biophysics experiments at SLAC. So the X-ray beams are coming in over here from the left, along what's shown here in this arrow. And this piping that's shown here is actually, there's some additional good robotics inside that, like some mirrors and some other things that enable the X-rays to kind of be focused and to essentially just have all of the qualities that we want out of our X-ray beam. There's this table here is actually where the sample is mounted. And then finally, we have a detector down here. And so what we're going to want is that the sample will collide with the X-ray beams, and we'll be able to measure all of the scattered waves on our detector over here. And so, we're finally at the experiment. We have all of the ultra bright X-rays delivered to our experiment. So now the next thing to do is discuss kind of how we deliver the sample so that it will collide with the X-rays, the ultra bright X-rays that we've just delivered, in order for the next part of our experiment to proceed. So I guess when I mentioned the Rosalind Franklin experiment, one thing I mentioned is that she had to have many kind of DNA molecules in parallel to one another. And actually the same principle applies here. Generally speaking, individual proteins are actually a little bit too small and don't scatter strongly enough for us to actually kind of measure the results of these scattering experiments. So one of the most effective strategies we have to overcome the fact that we get very, very little signal, almost too little to measure from a single protein, is to actually crystallize them. So that's part of sample preparation and delivery. And the reason it works to crystallize them in terms of actually getting more signal is because there's what's called constructive interference between the scattered waves. And this occurs when we have a crystal. So to start off with, a crystal is essentially any object where we have kind of translational periodicity. So in this example here, if you look at these gray spheres, you can see if you move any of the gray spheres over by 1D and either to the left, to the right, up or down, then what you'll see is you'll be in the exact same place again, because there's this translational symmetry. You can move by D and then it looks the same. So this is kind of a diagram of a crystal, and what's special about crystals is that when we illuminate them with some sort of wave radiation, and that's what X-rays are, then what we'll find is that in certain directions, the waves will actually line up. So as they scatter off, as they scatter off this crystal, these two waves, as you can see, the kind of the peaks of the waves and the troughs will line up. And the effect of that for, those of you who have taken some physics and seen kind of the effect of adding waves is what's called constructive interference. So instead of having kind of the amplitude of just a single wave, we have, double the amplitude because we have two waves scattering in parallel. And so it'll look like it's, a much larger wave as we get further and further away. And so the effect of the crystal is, instead of just having 2 rows of these spheres, we have, billions of rows of these spheres. With that, we can actually, get a lot of constructive interference such that instead of just having, twice the amplitude, we actually have, billions of times the amplitude. And so we actually get a really good signal by packing our proteins into a crystal. And what's, I think one of the crystals that we encounter somewhat frequently in everyday life is actually sodium chloride or table salt. But what's really cool is that proteins can form crystals too. And let me see if I can show this video. Okay, so this video was actually produced by NASA because there has been some interest in seeing how, whether proteins crystallize in space differently than they do when there's gravity here on Earth. And so what's really cool, let's see, it's 47, here it is. So what's really cool is if you have a bunch of proteins together and you have them in a very saturated solution, so you have a really large, high concentration of proteins together and you add in some salt, often they'll just spontaneously crystallize. So what's shown here, these yellow guys are both proteins, they're about to turn purple. And as you can see, this is kind of a schematic of them all crystallizing. They just spontaneously form this kind of translationally symmetric crystal where they just they just link on to one another, and then just the crystal begins to expand and expand. And then what's shown in this video down here is actually a movie of real lysozyme crystals. So lysozyme is a protein that crystallizes very well. And what's cool about this particular movie, it's sped up a little bit, but this movie actually would occur over the span of hours. You can watch these lysozyme crystals grow if you look at them underneath a light microscope. And so as you can see here, we just had, all that's in this drop is a lot of really concentrated protein and some salt. And you can see these are protein crystals just spontaneously growing. And in this particular case, they actually get quite large, as you can see here. And so again, what these protein crystals are, proteins kind of arranged in some sort of translationally symmetric way. And then, it's very effective for us as a tool in order to get signal amplification when we place them in front of an X-ray beam. So I guess one thing I wanted to mention is that this will have an effect in terms of what we actually observe downstream. Similar to the way Rosalind Franklin had her photographic film. Today we use kind of something different downstream. We use what's called a detector, and we get these diffraction images. And so if we look at our detector, if we just had a single protein, then we'd observe some sort of continuous signal. But the signal actually isn't strong enough for us to actually measure it on, normally speaking. And so if we have a crystal, once again, as I mentioned earlier, we'll get this constructive interference. So we get all of these discrete spots, as you can see right here on our detector. And then on the right, what's shown here is an actual diffraction image from a real protein crystal. And as you can see, all of these spots actually contain information about our protein structure inside of them. And so ultimately, that's what we'll be analyzing at the end. And so, now we have a sense of, most frequently what we're going to be using is not individual proteins, but for sample preparation, we'll want to make them into a crystal. And so as part of delivering them to our experiment, what we're going to do is we're actually going to jet them. We're going to kind of jet them in some sort of solvent so that they collide with the X-ray beam. And ultimately, these X-ray beams are so bright and so powerful that the protein crystals will actually explode. They'll be completely damaged. We won't be able to use them again. But that's okay because we're continuously jetting more and more fresh sample across this X-ray beam. And even before they're destroyed, we're able to collect this diffraction image that we discussed on the previous slide, as you can see here with all these really bright spots that are related to the protein structure that we're ultimately most interested in understanding and figuring out from this experiment. So are there any questions so far? Okay, if not, I'll move on to kind of the third part of this experiment, which is the data analysis. It's essentially going from the diffraction images that we've just been talking about to an understanding of kind of protein biochemistry. What does the protein structure look like, and how are these proteins moving around, and how does that lead to them functioning inside cells and giving rise to the biology as we know it. And so I guess essentially, during these experiments, we collect thousands upon thousands of diffraction images. And as we collect more and more diffraction images, we actually have enough information. I won't be able to go into kind of the details of what this arrow implies. This arrow implies some kind of algorithms that we, perform on all of these diffraction images, because we know specifically how these spots are related to our protein structure. So we can apply these algorithms to all of our diffraction images, and ultimately what we'll get out is the final output will be something that's called an electron density map. And so the electron density map is shown here on the right with all of this blue mesh. As you can see, there are certain bulges in this electron density map, and those bulges actually correspond to where individual atoms are inside the protein. And that's ultimately what we're after. And the size of the bulge is related to kind of how heavy a particular atom or element is. And so hydrogen is very light, so you'll see almost a very small bulge or maybe nothing at all. And then something like sulfur, an element like sulfur is much, much heavier, so you'll see a much larger bulge. And then based on that information, we can actually figure out, where all of the atoms are located relative to one another. Because each of these bulges really corresponds to kind of an atom within the protein. And so ultimately, what we can do is we know the sequence of a protein, and we can try to thread it through this electron density map and figure out where each atom in the protein is located relative to one another within this electron density map. And so ultimately what we'll get is what's called an atomic model of the protein. So where all of the atoms of the protein are located in 3D space relative to one another. And so that sort of information is actually really valuable for many applications, one of which is drug discovery. So actually what the example I'm showing here is the electron density map and the atomic model for one of the proteins that's really critical for the COVID virus to infect us. So in this particular case, this COVID protein is actually an enzyme, which is called a protease. And so its function is to kind of chew up other proteins inside the cell. And in this particular case, this is a crystal structure, so an atomic model of this protease, which is bound to an inhibitor. It's actually the inhibitor that's in the drug Paxlovid. And so the inhibitor is shown in yellow here, and we can see how all of the protein atoms, which are shown in kind of the gray and the other colors down here, are interacting with this particular drug. And so what this drug essentially does is it, based on the information that we get from this experiment, we can tell that what the drug does is it binds to where catalysis actually occurs in this protein, and it inhibits it that way. And so you can think of it as, this is kind of the mouth where this enzyme tries to chew on other proteins. And this drug is essentially a massive rock that gets in there and is too hard for the protein to chew on, but it's also kind of too bulky for the protein to actually spit it out. And in that way, this COVID protein is actually inhibited by the drug Paxlovid. And so, having this information from these X-ray crystallography experiments can be really critical for our understanding of things like drug discovery. There's also what's really one of the really special things about X-ray free electron lasers, which is the technology we were discussing to perform this particular experiment, is that we can actually also essentially acquire enough data sets and acquire them in different conditions that we can actually stitch them together to make a molecular movie. And one of the ways we can do that, so if we go back to kind of the schematic down here, one of the parts of this schematic that I didn't really highlight earlier was kind of this laser, which is shown in this bottom left here. And so there are lots of enzymes that are actually light sensitive. And so, if you shine a light with them, it will cause them to undergo some sort of structural rearrangement and force them to perform catalysis. And so what's really special about a free electron laser experiment with crystallography is that we can actually have a laser timed, have a laser be delivered to the sample as well, even while we're delivering the X-rays. And what's really nice is we can get the timing to be incredibly precise. And in that way, we can collect, a sample before any laser pulse has hit it. And then we can, turn on the laser and then collect more data after the sample has been exposed to some laser. And we can do that in some sort of way where we look at, okay, so we start in the dark case, we turn on the laser, we look at the, how the sample has changed after it's been exposed to some light very briefly. And then we can look at it a little bit later and a little bit later, et cetera. And, based on that, we actually kind of can stitch all of these, this information together to construct a molecular movie of how catalysis is actually occurring. And so that's exactly what was done in kind of the data that I'm showing here. And so, this is what we're showing here is essentially akin to like as though we were watching dancers in a strobe light. We get these kind of frames that we know are related to each other over time, and we can watch this chemical reaction evolve in time as well. And so that's what's shown in this movie here. You can see the electron density, again, shown in the blue mesh, changing as a function of time. And by the time you get to 8 milliseconds, you can see it's in a completely different rearrangement. And so this let us actually see with atomic detail how this particular reaction was occurring. This reaction is actually how reaction inside a bacterial protein, which is how bacteria sense light. And it's actually the same exact chemical reaction that the proteins in our eyes use to sense light as well. And so, what's really special about these X-ray free electron lasers is we can get this sort of information about how, these chemical reactions are occurring in enormous detail. And we can assemble these molecular movies of how proteins are operating. And so I guess I wanted to conclude by showing this video that SLAC put together a few years ago that I think really kind of brings together all of the things that we've talked about today. So let's see if I can get it. Okay, so in this movie, we're going to start once again with kind of an aerial view of, first of all, we're going to zoom in from the United States onto the Bay Area, which is shown here. And we're going to zoom in specifically where SLAC is located in the South Bay. And as you can see, this cluster of buildings is where the experiments happen. And then this long line here is the particle accelerator. And in order to construct the free electron laser, which is the really exciting piece of equipment that we use now for some biophysics experiments, they actually had to extend the particle accelerator, which is shown here in kind of the orange. It's this long light, and it ends at the experimental halls where the hutches are, where we actually perform the experiments. And so this little video is showing, animation is showing the X-rays being delivered to the experimental hall. And so what we're going to do is we're going to start at the other end of the particle accelerator where things begin in the first place. What I didn't have time to discuss earlier was how electrons are generated in the first place. And they're done so with this injector gun, which spits out these ultraviolet pulses, which are shown here in red. The pulses will hit this copper tube, which is shown back here, and that will force electrons to be generated. So these electrons are initially, these guys shown in blue are initially a little bit slow, but that's okay because we're going to get them up to speed, nearly the speed of light again. And so the first thing that happens is also something that I didn't have time to discuss, but essentially we put this little bend in the particle accelerator so that any electrons that are going too fast are slowed down just initially, because we want them all going the same pace before we really get them going very fast. And so that's what the chicane here is. But once we have them all going at the right speed, now we're going to start accelerating them all from that speed up to nearly the speed of light. And again, we're doing that by having all of these cavities made of superconducting metal with an electromagnetic field inside so that the electrons start going very, very fast. And so these electrons, again, they're the blue dots. They'll start zooming down the particle accelerator, down all of these cavities. We're getting them going quite fast now. And as you can see, it's a very long way of cavities. The electrons have quite a ways to go for us to get them up to speed. But at the end of the cavity section, this is actually another section where we just have some diagnostics to make sure that the electrons are of the right quality to begin emitting X-rays of the quality and characteristics that we want. But after this portion of the particle accelerator shown here, what we'll get to is the second part of the particle accelerator we discussed, which is where there are these alternating magnets. So here, these electrons, these blue dots, are going to start oscillating back and forth. And as they start oscillating, as you can see here, they're going to start emitting X-rays. And so the X-rays are this kind of gold cloud that you see forming right here. And the X-rays are just moving straight. They're not oscillating back and forth like the electrons are. So they start to overtake the electrons, which causes more and more X-rays to be emitted. And so, we have very fast electrons going back and forth, and we have these kind of X-rays following along that are now very, very bright. And so the X-rays again are these kind of golden, bright yellow things that are zipping along our particle accelerator. And so the final thing we want to do is we want to get rid of the electrons, which are on top of the X-rays. And we do that by applying a magnetic field at the very end, which causes the electrons to be shunted down to the electron dump. And the X-rays aren't affected by any sort of magnetic field, so they'll keep zipping along into the hutches where the experiments actually happen. And that's what's shown right here. So as it turns out, there are several hutches at SLAC. I think there are 7 or 8 of them right now. Not all of them are used for biophysics experiments, but some of them are. And actually, the one in this particular movie is one of the ones that's used for biophysics. And so what's shown right here is similar to the photo of the experimental hutch I showed earlier. As you can see, there's lots of robotics, lots of that help us with diagnostics. The beam actually enters in through the hutch, collides with the sample that we deliver to it, and we get these diffraction images on our detector, and that's the data that we're collecting and the data that we ultimately analyze. And if we collect enough of these diffraction images, we're able to kind of piece together how they're related to one another, and we're able to get back to the protein structure with atomic detail, and that's what we're really interested in. Yeah, so hopefully that kind of brings together everything. This is kind of, these experiments are, classic biophysics experiments that are really at the intersection between biology and physics, and they're a lot of fun to perform and kind of really interesting in terms of the information that we can extract from them. And so with that, that's everything that I was going to discuss, so I'd be happy to take any questions.
I have a question for you.
Mm-hmm.
So this is very interesting work that you've been doing, and it's really cool to look at, especially with this last video and with the X-ray lasers and stuff like that. But with all that being said, have you ever, was your plan always to do this type of work? Did you have any type of other interest in any other career paths?
Yeah, that's a great question. So I actually kind of got into this by accident. So I started off, when I initially started off doing research, I started off in cellular immunology because I was really fascinated with the way the immune system worked. And then after that, that led to me being interested in disease in general. And so after that, I spent a few summers at a biotech company called Genentech, where I was working in the small molecule drug discovery team. And I just realized that a lot of the questions I was really interested in could be addressed by biophysics. So, basically knowing protein structure and function, a lot of that, biophysics experiments are really critical for that. And so just over time, I gradually became kind of more interested in methods development to enable our understanding of disease. And so now I'm more on the methods development side, but I'm still really interested in disease. And I think I guess I personally have found it useful to kind of have both perspectives, to, have been more on the application side about, knowing what's important for the field, but then to also be on the methods development side to, being able to say, okay, I know what sort of experiments we want to perform. This is how we can perform them.
Right. I have one follow-up question. Because you mentioned about diseases, and there was this article that I read, this headline about scientists finding like old diseases from like that were frozen up from like thousands and thousands of years.
Mhm.
And they're apparently calling it a zombie viruses. And it's kind of scary to hear about that, but like if it does come up to that, how like diseases like that, would it be very challenging to like deal with it, or challenging to understand it, and just our effect on people?
Yeah, that's a great question. It is really concerning, these, one of the challenges with these so-called zombie diseases is that we don't have any protection against them, because they, like our immune systems are used to what we've seen before. That's why we often have some immunity to the flu or some other sorts of diseases. But, these are diseases that humans haven't seen for hundreds or thousands of years. And so we don't have natural immunity to them. But in the same way that, COVID was very challenging for everyone, and it had a huge cost in terms of human lives, and also all of our experiences. But science was able to come up with a solution with vaccines, with drugs that work against COVID. And so it'd probably be a similar strategy there, where we have the tools to be able to kind of figure out a vaccine or figure out drugs that work against these. So, and I think the COVID pandemic has really opened our eyes to the fact that we need to be more proactive in these situations. We need to be thinking about kind of vaccine development more generally, as opposed to trying to be reactive and just after the fact trying to respond to a virus that has spread pretty widely. So I think that, hopefully in the future that science will be, or that the community will be a little bit more on top of things in terms of kind of looking ahead and trying to anticipate what sorts of diseases might spread very rapidly. And it's possible that some of them won't spread very rapidly. It's possible that some of them just won't be that effective at jumping between different humans, or won't be, will be pretty mild in terms of the symptoms. It's just hard to know in advance.
I was wondering, like, what's the goal of making electrons in the particle accelerator?
Yeah, so the purpose of having electrons is because there are means to getting X-rays. So we can't just generate X-rays from scratch, particularly kind of X-rays of the really high intensity that we really want. And so the purpose of having the electrons is because they're the tool by which we can generate these incredibly bright X-rays. And so, yeah, otherwise, if there were a way to just get to the really bright X-rays without the electrons, we totally would have taken that route, because it would have simplified a lot of things. But unfortunately, there isn't an easy way to get to that quality X-ray without having electrons that are going very, very fast.
Oh, okay, thank you.
Mm-hmm.
Oh, one question. So, does analyzing the images of proteins, does that in any way help us, prevent like diseases, or give us more insight into, conditions like in the future? Do you get what I mean?
Yeah, so in this example here, for instance, this is actually, so we knew before the crystal structure that this was an effective drug, because there are other ways to determine whether drugs are effective. But this actually is useful information for us in the future, because as you might imagine, this COVID protein is probably going to mutate over time. It already has mutated a bit. And so, some of those mutations are designed so that this drug is no longer effective. But because we know the way this drug binds, we can say, okay, if it's been mutated in that way, then maybe, this protein atom and this part of the drug no longer interact. So we can change the drug slightly. We can think of ways to think about specific ways to change the drug to kind of overcome the way the virus protein is mutated. And there are loads of cases like that where, kind of analyzing these images, knowing specifically the structure of proteins is incredibly effective for things like drug discovery. It's also incredibly effective for things like designing new enzymes if we want, something that can help us with biodegradability or things like that. And so this information is kind of just generically very useful for a lot of applications. Does that answer your question?
Oh, yes, it does. Thank you.
Any other questions? I see there's, let's see here, there is another question about whether niobium was the only element that can be used for the accelerator. I think niobium has been the most effective. I know there are other metals that are superconducting, but I don't know about their other properties, like how hard they are, or what temperature you have to lower them to before they're superconducting. Cost also sometimes plays a role because some of these things are incredibly expensive, and so I'm not sure why niobium specifically was chosen. There are other superconducting metals out there, but I guess it was probably the most practical. So yeah, there was another question about whether there's synthetic transactinide super heavy elements produced at the particle accelerator at SLAC. There may be, to be honest, my focus has really been biophysics, and so I actually don't know much about kind of the other things that are going on at the particle accelerator, though actually I suspect that the answer is no. The particle accelerator is really to deliver kind of photons and x-rays. X-rays, which are photons. That's the whole purpose. We get electrons. So those are all, those are very light particles. I think, places that are more interested in super heavy elements are other national labs, like Los Alamos National Labs, which deals kind of with some of like the uranium stuff and other elements like that. SLAC doesn't do as much work on that side of things. And can this type of X-ray be used on humans? Yeah, so these X-rays are incredibly bright. And so, yeah, it'd be very dangerous to ever be exposed to them. We essentially, as soon as we turn the beam on, right before we turn the beam on, we evacuate the hutch and we make sure that there is no human inside the hutch before X-rays start to be delivered, because it could be very dangerous. In the same way that I mentioned that, when the X-ray collides with a protein sample, the crystal actually explodes. And so it's very harmful to biological samples. It'd be very harmful to humans as well. So you need like lower energy X-rays for the sort of imaging you do for bones and stuff. And then another question was, how would an enzyme be designed? Okay, so enzyme design is, so usually enzyme design starts from the enzyme we already know. And so often, actually, an example of this is there are enzymes that are used there are enzymes that are used when we wash our clothes, for instance, that try to break down some of the particles. And one thing we've done is to start with enzymes we know could break down certain things, and then just try to improve them, try to make them be more resistant to higher temperatures so that we can wash things at higher temperatures and still have the enzymes be functional, things like that. And sometimes we want to change what the enzyme is reactive to. So usually we'd start with an enzyme we know. We'd often start with the atomic structure, the atomic model of, how the protein is arranged. And then from there, we'd make guesses about, okay, I think if I mutate this residue, then it will have this effect on catalysis or on the other functions of the protein. And from there, you could slowly but surely kind of migrate your way towards a better enzyme with the qualities that you want. There's a second approach to doing that. Instead of directed evolution, or instead of kind of that directed approach, it's what's called directed evolution, where essentially you go through rounds of just kind of selecting for enzymes. You introduce mutations randomly, and then you select for enzymes with the best properties. And then you kind of do that in an iterative fashion. So, you've done one level, you have a slightly better enzyme, you'll do more mutations on that. Hopefully you'll find something that's slightly better, et cetera, until you get to the property you want. So, yeah, I hope that answered those questions, but I'd be happy to follow up on any of them if that didn't answer them satisfactorily. Are there any other questions? No.
Are there any more questions? You guys can unmute or put them in the chat. Okay, I think that's about it. I don't see any in the chat. So, well, thank you so much for your time today. Ms. Peck, I'm sure it was like as interesting for everyone as it was for me. I definitely enjoyed it. And thank you for attending, everyone. So I've posted a couple of links in the chat. So if you want to view the recording of this session, as well as the recordings of the other sessions throughout this week, please visit our YouTube channel, which I posted the link to in the chat. And in addition, if you're interested in submitting a project for the STEM media competition based on the topics that the researchers have presented about throughout this week, make sure to save that dev post link, as that is where the projects should be submitted. And if you are interested in any other future events, please fill out the third Google form. And if you have any questions, be sure to email us at inkandmetal5773@gmail.com, which I've also put in the chat. Or you can message us on the Discord server, and any one of us will be happy to respond. So thank you for coming, and thank you so much for your time once again.
Yeah, thanks everyone. Thanks for all the questions. Yeah, thanks for organizing this. I hope that it's been a good week for this. It seems like a really valuable experience. Oh, sorry. I think
I was on mute. Sorry, I was on mute. Yeah, it was definitely like a great experience for us as well as the attendees as well. So we're grateful for SLAC to work with us for this program. So would it be also possible to send over the recording so I can share it with the participants so they can refer to it as well later on?
Yeah, yeah, I'll figure out the, oh, here it is.